Chalo, let's start our session.
Yes,
So
ma'am.
look it here. In first chapter, we studied about the classification, taxonomy, right? Kingdom, phylum. Yes. So you have to identify the image, and you have to tell me these organisms are belong to so and so phylum. Do you know what they are? No, but you said now in your school this is already finished. You already completed this topic in your school.
Yes, ma'am.
Then
I didn't understand some topics in that the animal kingdom.
you didn't understand some topic, but at least you can identify an organism, whether it is mammal, birds,
Aquatic animals, ma'am.
aquatic animals. Chalo, that is good. Yes, they belongs to Porifera and Coelenterata. This is the sponge. This one is sponge. Okay, sponge is what? Which phylum? Porifera. This is Hydra. It belongs to which phylum? Cnidaria or Coelenterata. Okay, so in today's class we are going to discuss about the classification of Animalia. Okay, so here the kingdom Animalia is classified broadly into two groups, that is protozoa or the parazoa and metazoa. Okay, so the animal kingdom is divided into two sub-kingdoms. Okay, right now we are not talking about the phylum, we are talking about sub-kingdom, that is protozoa and metazoa. What is protozoa? Protozoa is also called as parazoa. So what is protozoa? What is parazoa then? Yes? No idea? Okay. So parazoa or protozoa are the unicellular animal. Unicellular animal. Why I am saying this unicellular animal? Because they don't have cell wall, because they don't have chloroplast, and they are eukaryotic. And this is the typical cell structure of animals, right? That's why all those organisms who are having such kind of cells, the body is made up of such kind of cells, they comes under the kingdom Animalia. Okay, this is the most primitive classification system, okay, for the kingdom Animalia. So all the protozoa comes under the kingdom Animalia, parazoa or protozoa. Parazoa. Zoa means animal. Okay. And they are unicellular. So unicellular animals, okay, they are comes under the parazoa or protozoa. As they don't have a tissues, as they don't have a organs or organ system, okay, they are completely made up of cells. So we have two types of parazoa: protozoa, which is completely unicellular, and parazoa. So the parazoa, it can be multicellular, and the example is sponge. Sponge is the multicellular cellular parazoa. Okay, whereas paramecium, amoeba, they are what? Protozoa, right? Now the next category, the multicellular, strictly multicellular animals. Okay, it is called as eumetazoa. Eu means true. Okay, so eumetazoa or metazoa, they are multicellular organisms, and the body are organized in a well-defined tissues, organs, organ system. Tissue level organization, organ level organization, and organ system level of organization. Okay, that means only and only Porifera belongs to the parazoa, rest other from the Cnidaria till the mammals or Mammalia all belongs to eumetazoa. Clear? Yes? Okay. So now let's talk about eumetazoa. Okay? And the not only the eumetazoa, because we are going to talk about the Porifera also, which is the parazoa. Now let's see the modern classification of kingdom Animalia, and this classification is based on the few categories, criteria, that is level of organization, then whether the coelom is present or not, the body symmetry, yes, and notochord, yes,
Yes, ma'am.
segmentation. So these are the criteria on the basis of which we are going to classify the whole kingdom of Animalia. So the kingdom Animalia classify the organisms on the basis of level of organization, the cellular and tissue. We have Porifera, which is having cellular level. Now the tissue level, again, there are n numbers of organisms which belongs to tissue level. So we have to check another criteria whether they are able to fulfill or not. So what are they? Presence or absence of coelom. If coelom is there in the body, then it will be one group. If the coelom, if the coelom is absent, then it will be one group. If coelom is present, then it will be another group. And the third group, where coelom is present in a pouches form, means they are mimicking like a coelom, but they are actually not coelom. So it is false coelom, pseudo coelom. Okay. Now the organism, the group with the pseudocoelom type of structure, there is only one group, that is nematodes, Eskelminthes. But there are huge numbers of animals which are having coelom or which are not having coelom. So first, let's categorize, let's classify the organism which is not having coelom. So how we will classify those organisms? On the basis of symmetry. Okay, so on the basis of symmetry, all the acoelomate organisms who is having radial symmetry in one group, all the acoelomate organisms which is having bilateral symmetry in another group, that is Coelenterata and Platyhelminthes. Okay, now let's see the coelomate organism. We know that there are n numbers of organisms, different types of organisms are there who is having true coelom. So how you will classify now this group? We will classify this group on the basis of germ layer cells. Okay, whether the mesodermal cells are formed from the single cell during the growth of embryo, or whether the endoderm cells are giving rise to coelom. So on the basis of that, the mesodermal cell is contributing in the growth of embryo, are formed from the single cell during the growth of embryo. We are having three types: Annelida, Arthropoda, and there is the organisms where endoderm give rise Phylum are again various types. So how you will categorize them? Then we will come to the next criteria, notochord. Whether they are having notochord or not. If they are not having notochord, they will be echinodermata. If they are having notochord, that means they are chordate. Now, again, we will classify the chordates into different groups on the basis of presence of notochord throughout the life, only during the embryonic stage, and in the adult stage. Are you getting me? So on the basis of that, we will classify the chordates. Clear? So now let's see our first phylum, and that is Porifera. Okay. So this is our phylum Porifera. What is Porifera? Porifera is commonly called as sponge. Okay, it is commonly called as sponge. And they are aquatic animals. They can survive only and only in the water, aquatic habitat. Okay. Some poriferas are sessile, solitary, or colonial. So, sessile means, what is the meaning of sessile? If the organism or anything is attached or fixed to particular substrate, means fixed position or fixed to substrate. Substratum, substrate. Okay. If they are fixed and cannot move, okay, we use the term sessile. Solitary or colonial. As you can see, n numbers of sponges are there. Okay. They are living as a colony in a group. Okay. This is not a individual. This is the branch of this particular main Porifera. Are you getting me? This is the branching. But when n numbers of different or the individual one, they are living together at one place. This is colonial, means they are making a colony. In a group they are living. Okay. So these are the characteristics. Now let's see the structural characteristics. Okay. As you can see in the body of the Porifera, they are n numbers of pores, right? So their body form is what? Porous. They have n numbers of pores. These pores, which is present laterally throughout the body, they are called as mouthlets ostia. Okay. This huge, large pore at the tip. Okay, at the tip. This is what? Osculum. This is only this much large pore is there in the body, and this is called osculum. This is only one opening. Okay. As you can see, laterally there are n numbers of pores are there, but osculum is only one. Ostia means n numbers. Are you getting me? Okay. So this mouthlet ostia is just like an inlet tube. Hello.
Yes, ma'am. Can you hear me, ma'am?
Yes. So this ostia is like an inlet, means they allows to enter something inside the body. Either the food, water, air. Okay. But osculum, the huge, large single opening, is for excretion, to excrete out something, means for exit. Are you getting me?
Yes, ma'am.
Okay. Now let's see another characteristic. As this is the body of Porifera, these are the ostias, okay, which allows to enter the things into the body, and this huge large opening at the tip is the osculum to exit the waste thing. Okay. As we know, Porifera is made up of loosely arranged n numbers of cells. That means the level of organization will be cellular level. Okay. As it is a cellular level, the organization is a cellular level. That means they are having two layers of germ cells. Yeah, germ layers. And that's why it is diploblastic. Okay, as they are sessile, they are fixed to particular space, to the substrate, they cannot move. Are you getting me? And because of that, they are not having appendages. Okay, as it is a completely made up of cells, no tissues are there, and that's why the body is not differentiated into different sections. And so they are not having head, they are not having alimentary canal, the neck, the trunk, the appendages, limbs are not there because of the cellular level of organization. Okay. The whole body, we know that it is made up of cells, right? Loosely arranged cells. That means they are having mainly the whole body wall. This whole body wall is made up of two layers. Okay, the outer one and the inner one. So the outer one, epidermis. This outer one is called as pinacoderm. Okay, the outer one is pinacoderm. And this pinacoderm, this epidermis is made up of pinacocytes. You know why they are called as a pinacocytes? Because they are having osteas, right? And sponge use this ostia for a drink purpose. Pina, right? Pani pina. In Hindi, it is called as pina. If any cell is drinking something, it is called pinacocytes. Okay. Pinosis. The osmosis, if because of the osmosis, something is getting enters into the body, so it is pinosis. Okay. So just like that, pinacoderm means to drink. Are you getting me? And why we are using to drink? Because they have ostia which use as an inlet holes. Yeah, inlet pores. Another one, the inner layer is choanocytes. This layer, inner layer, or the endodermis. Yeah, that is what choanoderm, and this choanoderm is made up of choanocytes. So this one is the choanocytes. Are you getting me? We can also say that pinacoderm is the dermal layer. This one is the dermal layer. Okay, whereas choanocytes, which is in present inner side of the body wall, it is called as gastral layer. Gastral layer. Why it is called as gastral layer? Because in the inner side, the choanoderm, they are having n numbers of cells and they are performing different activities. Are you getting me? Yeah? And that's why it is gastral layer. Clear? These two layers, choanoderm and pinacoderm, they don't have basement membrane. Lack of basement membrane. As they don't have basement membrane, then there must be the chance that both the layer will get stick to each other, right? No, because in between these two layers, they are having gelatinous matrix. Okay, this one, this yellow color, orange color structure, the orange one. Okay, this orange one is what? The gelatinous matrix. That means this is non-cellular part of the Porifera. Okay, this is what? Non-cellular part. Which part of the Porifera is non-cellular? Mesohyl. Okay, the name of this part is mesohyl. Clear? Or mesenchyme. Mesenchyme or mesohyl. It is what? It is interconnecting gelatinous matrix which is not made up of cells, but they are the gelatinous matrix that separates the two layers. Clear? Okay. Now, as we know that Porifera is completely made up of cells, there is no tissues. That means they will have different types of cells, right? Yeah. So now let's see the types of cells which is present in the Porifera. From the image, you can tell me some name of the Poriferian cells. Okay, in mesenchyme, this is the mesohyl, right? In mesohyl, you can see there are amoeba-like cell, right?
Yes,
Amoeba-like
ma'am.
cell. This free-moving amoeba-like cell is amoebocyte.
Side.
Okay, this amoebocyte will differentiate into the arachnocyte. Okay, and this arachnocyte is having a very special kind of property, that is totipotency. Totipotency. What is totipotency? If the single cell is having ability to develop the whole organism, okay, this property is called as totipotency, and that particular cell is called as totipotent. Here in tissue culture, you must have studied this particular term, totipotency. Yes? Hmm? Hmm. Okay. So till now it is clear to you? Okay. As the archaeocytes, not archaeocytes, archaeocytes. Okay, this archaeocyte is what? Totipotent cell. So this cell will transform, or they have the ability to transform into different other types of cells, that is sclerocytes. Okay, this totipotent amoebocyte can transform into sclerocytes, into the spongocytes or coelencytes. Okay, they have the ability to transform into the sclerocytes or sclerocytes and what? Just now I told you spongocyte. Okay, done. And the main function of the archaeocyte or amoebocyte, the normal amoebocyte, the function is the transportation of nutrients. Okay, this one is having the function of transportation of nutrient. Okay, and this one is having the role in reproduction. Are you getting me? Yes? Now let's see the sclerocytes. This sclerocytes secretes, this sclerocytes will secrete, as the name itself suggesting, something related to cilia, right? So the sclerocytes will secrete calcareous spicules or siliceous spicules. This one, okay? Calcareous or siliceous spicule. Okay, so basically this sclerocytes form this spicule. Okay, spongocyte. Spongocyte secretes spongine. It secretes spongine fiber. So basically this spicule is a skeleton for the Porifera. But in some of the Porifera, along with the spicules, spongine fiber also acts like a skeleton. Are you getting me? That means we can say that sclerocytes and spongocytes secrete something which is responsible for the formation of skeleton. Yes, endoskeleton. Hello, Aarti. Are you there? So this sclerocytes once secrete the calcareous and siliceous spicules, okay, which remain inside the mesohyle, and then slowly it will turn into the more hard and permanent structure, and now we can call it as a spicule. So a spicule is a stiff rod or spike-like structure which is made up of calcium carbonate or calcium carbonate or silica. Yes. Okay. As spicule is a skeleton for the poriferas, that means they give structure and helps in defense. Structure and defense. Please write it down. First, write amoebocyte. Now make an arrow. Amoebocyte will make transform into the which cell? Archaeocytes, spongocyte, and sclerocytes. Now make an arrow. Sclerocyte will secrete calcareous and siliceous substance, and then it will form spicules. Spicules provide structure and defense, protection, not defense, protection. Spongocytes form spongine fiber, which acts like a skeleton. Archaeocyte helps in reproduction. Amoebocyte, if the cell will remain as a amoebocyte, so this amoebocyte will help in the transportation of nutrients. Clear? Now let's see another cells. For example, choanocytes. Okay, choanoderm is having choanocytes. So this head and tail like structure is choanocyte. Okay, so which one is choanocyte? This one. Okay, this choanocyte is also called as collar cell. Okay, as you can see, there is a head-like structure, and from there, n numbers of flagellas are coming out. Yeah, that means collar cells are flagellated cells, and they help in digestion. So, which one is the digestive system for Porifera? Choanocytes or collar cells. Okay, digestion. Who is doing digestion here? Collar cells. Now let's see porocyte. As you can see, there is a pump or the channel or the pathway like structure. This pathway is actually behind these pores. Okay, the cells behind the pores who is making the channel for the passage, channel for the inlet of substance, the water, nutrients, gases. This cell is called as porocyte because they are making pores. Are you getting me? So this cell is specially for the passage of water. Write it down. Passage of water. Now let's see the epidermis, orange color wala cell, pino or pinocytes. This pino or pinocytes make the outer protective layer. Only the function is outer protective layer. Clear? Some of the pinocytes, it get modified into pinacocyte, sorry, not pino, pinacocyte. This pinacocyte transformed into special type of cell, and that is mycocyte. Mycocyte. Okay. Now, what is the function? Why it is there? Yeah. Pinacocyte, they are making the outer layer, right? And the outer layer of whole body is having same pinacocyte. Okay. Then what is there who is forming or making the outer layer epidermis for this huge large opening osculum? The modified pinacocytes. Okay, so this pinacocytes modified into mycocytes, and this mycocyte will form epidermis or the outer layer of the osculum. So basically, what will be the function? It will control the size of osculum. It will make the it is responsible for the opening of the osculum, opening of the pores. Clear? Yes, different cells are clear to you. Sure. Okay, now the very most important thing. The structure of sponge is like outer layer and nothing is there inside. The hollow structure, right? This hollow structure, let's move to the nothing is there, no cellular structure is there, right? So this is the cavity which is called as spongocele. Spongocele is what? Cavity. Okay, this spongocele is a large cavity, okay, which is actually lined or covered by the collar cells. Are you getting me? And this spongocele is actually responsible to collect the thing, whatever things are coming from outside through the pores, ostia. Okay, after the metabolism, all the excreta will again come to the spongocele, and now the excreta will come out from the osculum with the help of spongocele only. Okay, So spongiosale is the internal body cavity, the large cavity which is lined by collar cells. Okay. The structure is clear to you? Okay. Now, let's see, as we know that the Porifera is having the porous body, right? All over the body, they are having pores. And because of the pores only, they are able to get the nutrients, water, they are able to perform the respiration, digestion, excretion, everything, right? So this movement, because they are aquatic, they don't have specific mouth, they don't have specific organ system. Are you getting me? So how they will regulate the incoming and outgoing with the help of n numbers of pores, ostia as well as osculum, and with the help of water, because they are present in which medium? Water, right? So on the basis of how the water is coming out, how the excretion is taking place, they are having different canal system. Okay? Canal system. This canal system helps the porifera in nutrition, respiration, excretion. Okay? So on the basis of their structure, the folding of inner wall, basically how the inner wall, coanoderm, inner wall, inner layer, how they are folded. On the basis of their foldings, there are three types of canal system. Canal system is classification, or the types of canal system is based on the folding of inner wall. Okay, so we have three types. Type ascon or asconoid. Asconoid. Okay. Type cycon or cyconoid. Type lycon or type leucon or leuconoid. Okay, as you can see, the asconoid is the simplest canal. There is no such foldings, right? And the example is leucosolania. Leucosolania. And Olinthus. These are the example of asconoid. Now the cyconoid type. Cycon. Cycon is a type of fungi, porifera or cypha. Okay. Next is leuconoid. Leuconoid is the most complex but very efficient canal system. Are you getting me? This leucone is a very complex, as you can see here, the inner wall, this inner layer are folded in such a way that they are making small pouches, right? And each pouch, each structure is connected with the layers. So here are the porocytes. See, porocytes. Are you getting me? So though they are having a very complex structure, but the most efficient system is leuconoid. Whereas you can see here in the cycon, they are less complex, more complex than the ascon and lesser than the leucon. But as you can see, they are arranged below the pinacoderm, yes, and they are, one porocyte is connected to the different way, means one porocyte is connected to this one. So efficiently they are not able to properly, they are not able to equally, they are not able to distribute the water, the nutrients. Are you getting me? Yes, Aarti. And that's why it is said that most efficient one is leuconoid. Okay, what are the examples? The best example is spongilla. And euspongia. Euspongia. These are the examples of different canal system. Okay, and the examples are very important. Now let's see the physiology and other life processes of Porifera. So first we have a reproduction. Here the Porifera, they reproduce asexually as well as sexually. Okay, asexually they can reproduce through the spatial structure gamete. This is the structure of gamete. So this one is the structure of your sponge. Okay, here you can see in the mesohyle, this one is the mesohyle where these spicules are there, right? Mesohyle, the amebiocyte transform into the spicules and transform into the archaeocytes. Okay, archaeocytes. So how they are arranged? The outer layer of mesohyle will have a spicules and the inner layer will have a archaeocytes. Okay, and this one is what? This one is the micropyle, just like in a seed, for the incoming and outgoing of nutrients, water. Okay, and they cover the whole area in such a way that they are making a spatial structure like thing. Okay, and here they start forming the internal bud. Here the cells of archaeocytes will start forming these small internal buds, and now these buds will coming out from here, the micropyle, and they will germinate and develops into the whole organism. This is the gamete, by the process of gamete. Okay, fragmentation, what you have studied till now about the fragmentation. One of the organism will uh, get split into the fragments. Okay, and one of the fragments will come and once it will come into the contact with the substrate, as this is the sessile, so they need substrate. In a water current they cannot survive. Okay, so they need substrate. So once one of the fragment will come in contact with the substrate, now this part, okay, will survive as an individual. So this is what fragmentation. Clear? Now let's see the sexual mean. Okay, in the sexual mean, as we know that archaeocyte is responsible for the reproduction. Yes, so here in the sexual reproduction, we know the first we need to know that it is a hermaphrodite. Hermaphrodite means it is a bisexual. Okay, in the same sponge, the body will have male as well as female body. Okay, so how the sex cells, sperm and ova get developed? It will develop from the archaeocytes or choanocytes. Archaeocyte, which archaeocyte? The undifferentiated one. Amoebocytes will transform into the archaeocyte. Okay. Now this archaeocyte will not differentiate into another structure, but this archaeocyte will develop the male and female gametes. Or the choanocyte, the collar cells, this one will develop the male or female cells. Okay. Then what will happen? They will release the cells into the spongiocele where the fertilization will take place. That means fertilization is what? Internal. Okay. Here it is saying oviparous fertilization. What does that mean, oviparous fertilization? This is not a general one. In some of the sponges, they shows oviparous fertilization, where the sperm and the egg, they fuse together in a water, in an open area. That means we can say it is an external one. External fertilization. Yes? Are you getting me? Okay, so first they will release the cells into the outer environment where the fertilization takes place. Whereas in some sponges, there is viviparous fertilization, which means internal, inside. I said there is an internal fertilization and external fertilization. So that internal one is viviparous. Clear? After the fertilization, what will happen? Zygote will undergo the process of cleavage, right? The fused structure is what? Zygote. So zygote will undergo the process of cleavage. Which type of cleavage? Holoblastic cleavage. What is holoblastic cleavage? What is holoblastic cleavage? When the zygote will complete the cell division. This zygote will completely split into two daughter cells, divide into two daughter cells completely. This type of cleavage is called as holoblastic. Clear? After the cleavage, there will be a embryo development. Yes? So what will happen? After this, the zygote will undergo the process of cleavage and they will form larva. Okay. And then larva, if it is a external fertilization, so larva will be there in the external environment only. Okay. If it is internal environment, so the zygote will come out through the spongiocele. Okay. And then the larva will develop into the external environment. Okay. Yes. So this is all about the reproduction. Till now it is clear to you? Okay. Now let's see the classification of Porifera. We used to classify the Porifera phylum into 3 classes on the basis of skeleton. Okay. If the skeleton is made up of calcium carbonate. What is the skeleton here? Skeleton here, spicule. If the spicule is made up of calcium carbonate, then all the organisms will come under the class Calcaria. Okay, Calcaria means calcium. Yes, and the examples are cycon, then leucosolania, then grantia. These are the examples of Calcaria class. Means spicules are having calcium carbonate. Next is the hexatinellida. Hexatinellida. Okay, which means the spicules will be made up of silica, siliceous. If the spicules are made up of silica, then such kind of porifera will comes under the class hexatinellida. Okay, hexatinellida. And the examples are hexatinellida examples are Euplectella, Euplectella. Sorry, it's U, not Euplectella. Another example is Hyaloma. These are the examples of hexatinellida. Okay. Next is Demospongia. Demospongia. Here the demo means the word demos, which means frame. Okay. If the skeleton is in such a way that it will look like a frame, okay, then such type of sponge comes under the group Demospongia. So here the spicules are not there, but they are having spongine fiber from the sponge. Remember? So they have spongine fiber as a skeleton. And the examples are Spongilla, okay, then Cleona, Euspongia, Euspongia. These are the examples of Demospongia. Is it clear to you, RP? Porifera? There are various types of porifera or the sponges which have different names and which have a different importance. Okay. So we have one sponge which is also called as, let me show you the image. Sponge, Venus flower basket. This sponge is called as Venus flower basket. In Japan, this sponge is used as a wedding gift. Okay, bouquet. See. What is the name of this sponge? Venus flower basket. And let me show you how they look like in their actual habitat. This is what? This is Venus flower basket. And this is actually what? This one. Siliceous. Okay. Next, another example is a bath sponge that we use for bathing purpose. Bath sponge. This bath sponge is nothing but the Euspongia. 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 Okay, bath sponge is what? Euspongia. Then we have another type of sponge like freshwater sponge, that is spongilla. Spongilla is a freshwater sponge. Then Cleona, that is called boring sponge. We have one more type of sponge, that is boring sponge, Cleona. But it's called boring. वो the psychological wala boring nahi, okay, mental wala boring nahi. This boring means to bore, to penetrate something, just like a borer, wood borer, the bird. The same way, this Cleona sponge has the ability to penetrate the rock, okay, and that's why it is called as boring sponge. Clear? So this is all about the Porifera. Now let's see the next phylum, that is Cnidaria or Coelenterata. This Cnidaria, the name Cnidaria, the word comes from the Cnide. Okay, and the meaning of this Cnide is sting or the nettle. Okay, sting cell means if the organism is having sting cell, okay, they are Cnide. If the organism is having Cnide, so let's call them Cnidaria. Okay, Cnide bearing organism are Cnidaria. Coelenterata. Coelenterata, why it is called as Coelenterata? Why they are called as Coelenterata? We know that it is Cnidaria, but why it is called Coelenterata? Because from this phylum, now the level of organization will bit more advanced, the tissue level. So there was a chance that body will have a germ layer. Yes, they start showing or mimic, or it will look like they are having body cavity. It is not actually there. Okay, and that's why it is Coelenterata. So now let's see the general characteristic of Coelenterata. They are mostly marine. Sponge, they are mostly marine, but some are freshwater, like Spongilla is the freshwater sponge. Yes? So Coelenterata, most are the marine, few are the freshwater. Okay? Some are sessile, means fixed to the substrate. Some are free floating. Yes. So the freshwater Coelenterata is what? Hydra. Okay. Few examples of freshwater is Hydra. Rest others are marine. Okay. They are some are sessile, some can move freely. Yes, sessile can move freely, and they have a special kind of cell, which is what? Special kind of cell, which is yes, Aarti.
Cnidocytes, ma'am.
Cnidocytes. See the silent word. Okay. So because of the cnidocytes, the name is Cnidaria. Okay, which is a special cell which is having a special kind of stinging organelle, that is nematocytes. Stinging organelle, that is nematocytes. Nematocytes. Okay, or this one is also called as cnidoblast or nematocytes or cyst. Nematocytes, nematocysts. Okay, and sorry, or blast, cnidoblast, cnidoblast. Okay, so this is the uniqueness or unique feature of the Cnidaria. Coelenterata or Cnidaria is having two different types of body form. One is medusa and another one is the polyp. Okay, the medusa, which looks like an umbrella, okay, looks like umbrella, and the tentacles are uh, towards the downward side. Okay, and the polyp is like something is here where the hair like, they have a hair like thing. Okay, the straight projecting towards the opposite side or the straight form, whereas in the medusa the tentacles are towards the downward, means the opposite form. Are you getting me? So we have a two types of Cnidaria, polyps and medusa. This one is the polyp and this one is the medusa. Okay, now let's see the structure. These small cells, stinging cells, are what? These stinging cells are cnidocytes, cnidoblasts. Yes, yes. And why they have this cnidoblast or the cnidocytes? To catch the prey, right? Yes. As you can see, they have a two layers of cells, right? When they have a two layers of cells, that means it is diploblastic, yes, ectoderm and endoderm. They don't have mesoderm. Instead of that, they have a mesoglia. Okay. Here in the polyp, as you can see, the mouth is present at the bottom, somewhere around here, at the base. Whereas in the polyp, the mouth is present at the dorsal side, upper side, exposed side. Okay. So here it is the mouth in polyp, and here the mouth which is present internally or at the base or ventrally in medusa. Yes. Okay. Now let's see the symmetry. If you will cut the Coelenterata from the center axis by any plane, you will get n numbers of equal halves, right? From any plane. That means it is radial. The symmetry is radial. Are you getting me? Okay, some of the Coelenterata can survive, can complete their whole life cycle either in the medusa or polyp form, whereas some can interchange. Some at one stage of life they are medusa or the polyp, and then in another phase of life they change their body form. Okay? When any Coelenterata shows such kind of property, they are called as a polymorph. Polymorph, or this type of property is called as polymorphism, because they are having more than one form of physical appearance, morphology. Okay, and for those who are having polymorphism, they shows alternation of generation, that is metagenesis. Clear? Okay, what is the level of organization of Coelenterata? Cellular level or tissue level?
Tissue level, ma'am.
Tissue level of organization they have. So now let's see the tissue level organization, the structure. Okay, the whole body wall of the Coelenterata is divided into two layers: epidermis, the outer one, okay, the outer one, and the inner side, that is gastrodermis. Okay, in between the epidermis and gastrodermis, there is a layers. Okay, there is a layer of which consists of cells. This layer consists of cells, and especially which cell? Nerve cell. This layer is called mesoglia. Okay, so epidermis, the epidermal layer of the Coelenterata, it consists the special type of structure, that is cnidoblast or nematoblast. Okay, and the at the opening, at the tip of the cnidoblast, they have a special structure which is called sensory cells. So from here they get sense, okay, the food is there, and they will catch. The tentacles will move like this and they will catch the food. Are you getting me? Then what will happen? From the cnidoblast, the food will move to the next cell for the purpose of digestion and absorption and the storage. Okay, from here it will reach to the gastrodermis. Okay, they have one more type of cells at the epidermis, that is interstitial cells. Okay, what is the interstitial? What is the role of interstitial cell? Yes.
They capture food, ma'am.
Interstitial cells can capture food. Hello.
Yes, ma'am.
Yes. So this interstitial cells are, wait, yes, this interstitial cell, okay, which is present in the epidermis, this cell is actually the reserve cell. Okay, and these cells will then transform into the nerve cells. Okay, so you can see here in the gastrodermis also they are having some of the interstitial cells. Yeah, so these interstitial cells are the totipotent cell. Okay, they have the ability for the reproduction also. Okay, plus they get transformed into the nerve cells also. Clear? Now let's see the gastrodermis. The gastrodermis are having the layer of the cells, that is endotheliomuscular cell. So this endotheliomuscular cells, what they will do? They are the nutritive cells. They provide nutrition. So once they catch the prey, it will reach to the endotheliomuscular cell, from where this cell will provide or distribute the nutrition to the different parts of the body. Then again they have an interstitial cell, okay, in between these pillar like cells, okay, either the glandular or the endothelial cells, okay, endotheliomuscular cells. Then they have some nerve cells also, and they have a sensory cells, okay, sensory cells. Some of the endotheliomuscular cells are transformed or start to forming a pseudopodia like structure to circulate, to provide the nutrients internally inside the body, okay, plus to get the excreta from the other part of the body, and then it will diffuse to the one cell to next cell and then to the outer environment. Clear? Yes. So this is the structure of sea anemone Coelenterata. Okay. the whole layer of the body, this one, these are the tentacles, okay, these are the body. So whole body is having two layer of tissues, epidermis and gastrodermis. So this one, what you can see, the brown color one is the epidermis, and then internally they have a gastrodermis. Okay, this is the mouth. So what type of body form it is? Polyp or medusa? Is it polyp or medusa?
Polyp, ma'am.
Polyp. Okay. So the basic difference between polyp and medusa is like polyp will be cylindrical in shape, whereas medusa will be umbrella-like. Okay. Polyp is like some of are sessile and some are motile, but mostly the most of the organisms who is having polyp body form, they will be sessile. Just as you can see here, the sea anemone, it are sessile. Okay, they can make large colonies. They can be live separately in an isolated form, the solitary. But medusa, they are always free living because they are like this. They move like this. Okay, and they always live solitary. They cannot make colonies. Okay, so this Coelenterata is a bit advanced than the Porifera. They have tissue level of organization, not the organ level, and so the body is not at all, the cells are not at all differentiated, or the body is not having a different parts like head and appendages. Okay, they use the center part of, they are having the mouth. Okay, the end opening is the mouth. Okay, they have tentacles which is used as a limbs, as a appendages for the movement, for to catch the prey. Are you getting me? The body is like quite flexible, the tentacles are flexible. Okay, and they are hollow in nature. Clear. Now let's see the uh, internal physiological characteristic like digestion. Once the tentacles catch the prey and it provides food to the body or the mouth, what will happen? There will be further process, that is digestion, right? So they have two types of digestion, intercellular as well as intracellular. Intercellular means, intercellular means extracellular digestion. Intracellular means inside the cell, means more, more than two cells, okay, are involving for the digestion, then it will be extracellular. If only one cell, okay, digesting the particular nutrient, and it will be intracellular. So Coelenterata is having two different types of digestion, intercellular as well as intracellular. Okay, now let's see the respiration and excretion. Here the respiration and excretion takes place by the process of diffusion. Okay, as they are present in aquatics, so what happens, the gases, the oxygen present in the environment will diffuse to the plasma membrane, then to the cell, then to the other parts of the body. Or if they want to excrete the waste material, the same way they will excrete out through the diffusion process through the body surface. Okay, the main excretory material, the excretory matter, is the ammonia. Okay, the excretory matter is ammonia. They have a nervous system, the nerve cells, what you can see, what you have seen in the mesoglia and in gastrodermis, right? So the nerve system is in a diffused form. Okay, it is in a diffused form, and it is found in both the type of Coelenterata, whether the polyp or medusa. And they are like a network which is present throughout the layer, but in a not in a proper arranged form, not in a proper organization, but they are loosely arranged and making a network, loose fibers. Are you getting me? So, apart from the nerve cells, they are having sensory cells, which is present in the epidermis nearby the cnidoblast, right? Plus, in the gastrodermis nearby the endotheliomuscular cell. Yes. Now, the next property is reproduction. Porifera is what? Hermaphrodite, right? Whereas Coelenterata is unisexual. Okay, means one will be the male and another one will be the female. Okay, but it is not mandatory that all the Coelenterata, all the Cnidarian will be the unisexual. Some are there who is bisexual also. Okay, so they can reproduce asexually and sexually both. Yes. So how they can reproduce asexually, as we have seen in Hydra, through the process of budding. So whatever Coelenterata are having polyp body form, they can reproduce asexually through budding. But if they are having medusa body form, they will always reproduce sexually only. Okay. Okay. So they don't have the proper channel, proper structure by which they can release the what sex cells or the gametes. Okay. But they have the ability to produce gametes. Okay, they produce gametes, and either they will release, like these are the gonads, okay, where the gametes get produced, but they are not able to channelize that through via this. The gonads will go and it fuse, gametes will go and it will fuse with the female gamete. Okay, after the development of gametes in the gonad, they will bluntly scattered or spill or release either to the external environment or internal body structure. Are you getting me? And then when the male gamete will comes in a contact with the female, it will fuse and do the process, complete the process of fertilization. And that's why it is either the external or internal. Once the zygote will get formed, it will undergo the process of cleavage. Which type of cleavage? Holoblast. And then the cells will undergo the process of further development into the larva. Okay, so the zygote will turn into the larva. Okay, and this it will complete the process of reproduction. Clear? Now the classification of Cnidaria or Coelenterata. The classification of Cnidaria or Coelenterata is based on the body form. Okay, whether the polyp or medusa. But the body form, if the body form is dominant in their life stages or not. So the classification is based on the dominancy of body form. Based on dominancy or the dominance of medusa and polyp. Let's write it like this. Medusa or polyp form. On the basis of that, we have categorized, classified the whole phylum Cnidaria into clubozoa, sphaerozoa, anthozoa, and hydrozoa. Okay, so now let's see, the first one is Cubozoa. The Cubozoa is what? Jelly. Jelly-like. Not all the Cnidaria, most of them who is having medusa form, they are called as a jelly-like. Okay, because they have a umbrella-like and they have a very slippery structure. So it is jelly-like. Okay, so now let's see the Cubozoa. Cubozoa, why we use the term Cubozoa? Any specific reason for that? Yes, as you told me that you already studied.
Because they're in a structure of cub, ma'am.
Cub. Because they have a box-like structure. Okay, from both the sides they are making like this. Okay, and make the structure as are getting clubbed in such a form that they look like a box. Okay, so the examples or this structure is also called as box jelly because of the appearance. Box jelly. Okay, clear? Cubozoa, the box jelly. Any example of Cubozoa? Yes, any example of Cubozoa?
Jelly, ma'am.
Jelly. Yes. Okay. As this, the classification of Coelenterata is not that much important for your syllabus, but still I am telling you the example of Cubozoa is the Carukia. Carukia. Okay. Now the next category, next class is Cyphozoa. Here the cypho means, cypha means cup, not cub, cup. Okay, and that's why it is also called as the cup jelly. Okay, if they look like a moon, okay, so it will be called as moon jelly. If they are having a purple coloration, the pigmentation, so that type of cup jelly is called as purple jelly. Okay, and the example is Aurelia. Aurelia. Okay, this is the example of what? Cup jelly. Okay, next is Anthozoa. Anthozoa. Anthos means flower. As you can see, the sea anemone, it looks like a flower, right? The corals, it looks like a flower. So antho means flower like animal. Flower. Anthozoa, flower. Okay. And the examples are anemone, corals. Now the next one is hydrozoa. Hydrozoa, hydra means, hydros means water, water animal, hydra. And any other example of this? Obelia. Obelia. Are you getting bored, Aarti? Okay. Now the next phylum is Ctenophora. Ctenophora. So the Ctenophora is also called as sea gooseberry, comb jellies, sea walnut. Okay, these are the common or alternative name of Ctenophora. Okay. Okay. So now let's see the characteristic. Before moving to the general characteristic of Ctenophora, why we are calling it as a Ctenophora? Is there any specific reason? Yes. What is the meaning of teen? I always used to say, to know more about any term, first we need to split that. Okay, so let's split teen and phora. The C teen comes from the Greek word that is keteen. Okay, keteen, which means calm. Okay, and the next word is phora. This phora means bearing. Which means calm bearing. So calm bearing organisms are called as, are you able to understand what I'm trying to write? Huh. So calm bearing organisms comes under the group Ctenophora, or they are called as a Ctenophora. Okay, they have a very special kind of property to emit the light. Okay, only in the presence of sunlight. In the presence of sunlight, the comb plate, what they are having, as you can see here, these are the combs. Okay, this one is the body. Okay, and these are the comb plates. So in the presence of sunlight, these comb plates gives a lightning effect. Okay, and that is bioluminescence. And it looks like a rainbow, something are there, because rainbow is having 7 color band, right? And the Ctenophora is having 8 rows of comb plate, and that's why it looks like a rainbow. Are you getting me? Okay. So this effect, where the living organism is having ability to emit the light, this property is called as bioluminescence. Clear? Ctenophora are the strictly marine animals. You will not find any Ctenophora in fresh water. They are strictly marine animals. Okay? This is the diagram. Okay, this is the diagram of Ctenophora. The Ctenophora is having very soft and transparent structure, and that's why we used to call it as a jelly. Okay, if you can cut or split the whole body from the central axis through any plane, okay, you will be able to get the equal halves, and that's why it is biradial symmetry. Is it so? In the last class, I explained you what is biradial. Tell me that. Yes, Aarti.
Ma'am, if we cut at any axis, we get equal halves, ma'am.
Is the radial symmetry. And what is biradial? If we can divide the body into two similar halves, exact copy, two equal halves, when we are passing through the vertical plane, the condition is vertical. Make a star. Write it down. Biradial, vertical plane. Okay. As the Ctenophora, it somewhat look like a Cnidaria. The Cnidaria is also having a jelly-like structure, right? Ctenophora is also having a jelly-like structure. What makes the difference? Nematoblast, nidoblast, right? As the Ctenophora, they don't have nidoblast, and that's why they are also called as Cnidaria. Cnidaria. Because they look like a jelly. They both look like a jelly. But one is having nidoblast, another one is not having nidoblast. Okay, and that's why Ctenophora is called as Cnidaria, absence of nidoblast. Clear? Okay. They have a tissue level of body organization. They have a two layers, two germ layer, ectoderm and endoderm. So it is ectoderm, endoderm and mesoderm. Okay. So from here now the triploblastic arrangement are there. Okay. But here the mesoderm layer are not the prominent. Okay. They are having amoeboid, means the shape is not fixed, the line is not fixed, irregular lines, but still mesoderm are getting formed from this phylum. Okay, so it is triploblastic. Okay, as they have a amoeboid type of mesoderm, so it is called as coloblast. Clear? You can see here they have a complete. This one is the complete. Like this, throughout the body, they are having total 8 completes. The hair-like projection are the solid completes. On the body surface, on the surface of the complete, they are having cilia. And throughout the body, they are having 8 rows of ciliary complete. These completes, these combs helps in the locomotion like this. Okay. So now let's see the another characteristic, especially to the reproduction. The Ctenophora, okay, the Ctenophora are the bisexual. Okay, the single organism, they are having both the sexes. So what they will do, they will release the gametes and they will go for the fertilization, and after the fertilization the zygote will develop into the embryo, and that is to indirect type. There are two different types of development. One is direct and another one is indirect. Where the zygote is directly developed into the embryo, okay, or the larva, it is a direct type. When the organism are not directly developing, the zygote is not directly developing into the embryo, but through the different stages, so it is indirect type. Okay. So during the development, the zygote undergo the process of cleavage, that is holoblastic. So when the cell divides into two daughter cells, they will form two unequal daughter cells. Okay, unequal daughter cells. Okay, clear? And from here, they will develop the larva, as they have a sidipid larval stage. Okay, means, what does that mean? Sidipid larval stage means larva is not having that type of morphology that it will can directly convert, or it will directly transform into the adult one. They have a stages, okay, that they have to cross, and then they will develop into the individual. Clear? Now the classification of Ctenophora on the basis of the tentacles. Okay, the Ctenophora is divided into two classes. This classification is based on tentacles. Okay, there are two different classes, Tentaculata and Nuda. So Tentaculata are those organism or the class of those organisms who is having tentacles and absence of tentacles. They are having tentacles, and Nuda, they don't have tentacles. Okay. The examples of tentaculata is the Hormifora. Example is Hormifora. And the example of Nuda is Beroi. Okay. And this image, it belongs to, what is the name of this organism? The name of this Ctenophora is Pleurobrachia. Pleurobrachia. Okay. Whenever they will ask the question about the Ctenophora, most of the time they give they take the example of Pleurobrachia. Okay. So Pleurobrachia, let's see the structure. They have an anal pore. They have a mouth. Okay. So the tentacles they use for the movement only. Okay. For the ingestion, they use mouth. Okay. With the help of mouth, they catch the food, and after the completion of digestion, there will be an excretion. So it is through the anal pore. Okay. So as I told you that some are with the tentacles, some are without tentacles. So this Pleurobrachia, as you can see, they are having tentacles without nidoblast. Two tentacles are there, right? And they have a 1, 2, 3, 4, 5, 6, 7, 8 ciliated complex. Throughout the body, they are having small finger-like projection. Clear? So this is all about Ctenophora. Now let's see some questions. In case of poriferians, the spongocoel is lined with the flagellated cell in porifera. What is the flagellated cell here? Always remember, in the porifera, only one cell is there which is flagellated. Only one type of cell is there which is flagellated, that is choanocytes or collar cell. Okay, so correct answer will be choanocytes. Clear? Now let's see another question. Which of the following cells are present only in epidermis of Hydra? Interstitial cells, nidoblasts, sensory cells, germ cells. Interstitial cells were in the epidermis as well as gastrodermis, right? Sensory cells in the epidermis and gastrodermis, right? Nidoblast was there only in epidermis. So first will be the 2. So either B or A. Oh, sorry, either B or C. What will be the answer? Do they have a germ cells?
A, ma'am. B or
A?
C?
Yes, which one? B or C? B, sure. What about the germ cells? Let's see. So this one is what? This is the epidermis. This one is the gastrodermis. Okay. In epidermis, some of are having nidoblast, right? Right. But they are having one more type of cells that transform or that differentiate into the germ cell. And what are they? Interstitial cells that are totipotent cell. Okay, as I told you, only the interstitial cell which is present in epidermis, it will only responsible for the formation, responsible for the reproduction, not the interstitial of gastrodermis. Okay, that means interstitial of in the epidermis will form germ cell, right? And that means the correct answer will be option B and 4. Is option 2 and 4, and that is this one. Yes. Phylum Porifera is divided into two classes: Calcaria, Hexatenilida, Desmospongia, or Sarcodina, Sporozoa ciliata, Calcaria, Hexatenilida, Sarcodina, Mastigophora, Despongia. What will be the answer?
First one, A, ma'am.
A, yes. Ctenophora is nearest to Coelenterata, Porifera, Arthropoda, Annelida.
Porifera, ma'am.
How Porifera? Coelenterata is next to the, Ctenophora is next to the Coelenterata, right? I told you Ctenophora is also called as Cnidaria because of absence of nidoblast. But these two have a tentacles, right? So which one will be the answer? Which one will be the answer? They both have tissue level organization. They both have tentacles. Only the difference, one is having nidoblast, another one does not have. So what will be the answer?
A.
A. So in today's class, we have learnt about the Porifera, Coelenterata, and Ctenophora. Okay. Now let's see the class assessment. Okay, in the last class assessment, did you completed your? Did you submit your all the questions? How many questions were correct?
Yes, ma'am. I completed, ma'am.
And how many were correct?
Marks, ma'am.
Yeah,
9, ma'am.
9. And what about the 10th one?
I didn't check the answers, which is right, wrong, ma'am.
Okay,
Only
but
I checked the totals, ma'am.
okay, but did you remember the question?